Hello everyone, welcome to Aakash Telugu YouTube channel. ఈ రోజు మనం Recombinant DNA Technology లో కొన్ని స్టెప్స్ గురించి మాట్లాడుకుందాం. So the first step is isolation of the genetic material. It is a process of extracting the pure DNA from the cells, removing the cell membranes, RNA, proteins and the other cellular components. So we know that in majority of the organisms DNA acts as the genetic material. So DNA is present inside the cells enclosed in the membranes. Now we need to break open the cell in order to isolate the DNA. For example, bacterial cell wall is made up of peptidoglycan. So which enzyme do we need to use? We need to use the enzyme lysozyme to degrade its cell wall. Similarly, plant cell wall is made up of cellulose. So we use the enzyme cellulase, and fungal cell wall is made up of chitin. So we use the enzyme chitinase. Along with DNA, we have RNA and the surrounding histone proteins. Now, to remove RNA, we use RNase, that is ribonuclease, and to remove the proteins, we use proteases. Now, we obtain the DNA in its pure form, but we need to precipitate the DNA. DNA precipitation is done by adding chilled ethanol. Now, these DNA fragments will be seen as tiny thread-like structures in the suspension. The process of collecting these threads with the help of a glass rod is called as spooling. So, we are treating with ribonuclease to digest the RNA. We are using proteases to digest the proteins. We add chilled ethanol and the DNA is collected with the help of a glass rod, and this step is called as spooling. Now, the DNA is cut into fragments by the action of restriction enzymes. Restriction enzymes are the molecular scissors which help in cutting the DNA into fragments. So, we need to take a reaction tube, add DNA, add restriction enzymes and buffer to maintain the pH and distilled water as the solvent, and we need to incubate it at 37 degrees for the action of restriction enzyme to be done. To check the progression of restriction enzyme digestion, we perform gel electrophoresis. Gel electrophoresis is a technique where we separate the DNA fragments based on their size. Now, the most commonly used matrix is agarose. Agarose is a polymer which is obtained from a seaweed, so we call it as agarose gel electrophoresis. Now, we'll see the technique of agarose gel electrophoresis. So, first we take a casting tray. Into the casting tray, we pour the agarose solution. How is the agarose solution prepared? We need to take agarose powder, buffer and add the agarose powder into the buffer and boil it till the agarose powder completely dissolves, so that we will obtain the agarose solution. This agarose solution is poured into the casting tray and we insert the structure called as a comb. What is the function of a comb? It helps in creating depressions called as the wells where we load our DNA sample. Now, we allow it to solidify for some time. After some time, after the gel gets solidified, we remove the comb and we place the gel in an electrophoretic chamber and we add the buffer solution in it and we apply electric field. We know that DNA is negatively charged because of the presence of phosphate group. So since DNA is negatively charged, when electric field is applied, it starts migrating towards the positively charged anode. So care should be taken that well should be towards the cathode. Now, when electric field is applied, the fragments start moving according to their size. Larger fragments, they move slower because it is difficult for them to pass through the porous nature of the gel, so they remain close to the wells. While the smaller fragments, they migrate faster because it is easier for them to cross through the porous nature of the gel. So, larger fragments will be closer to the cathode and they will be closer to the wells, while the smaller fragments, they will be away from the wells and they will be closer to the anode. So, what is the principle here? Since DNA is negatively charged because of the presence of the phosphate groups, it starts moving towards the positively charged anode. The smaller fragments, they will move away from the wells and they will be closer towards the anode, while the larger fragments, they will be closer to the wells and they will be closer towards the cathode. Now, we directly cannot visualize the DNA under normal visible light. So, we need to add the stain, that is ethidium bromide, and we visualize the gel under the UV light. So, when you are visualizing it under the UV light, you see the DNA as bright orange colored fragments. So even though staining is important and visualizing it under the UV light is also important. When we are visualizing the gel under UV light, the DNA fragments are seen as dark orange colored bands. Now, the desired DNA fragments, the purified fragment will be ready to ligate with the vector. So, this is about agarose gel electrophoresis. So, if you talk about a glance of all these steps, the first step is cell lysis. We break open the cells. For bacterial cell, we use lysozyme. For plant cell, we use cellulase. For fungal cell, we use chitinase. To remove RNA, we use RNases. And to remove proteins, we use proteases. And why is chilled ethanol added? So that the DNA gets precipitated. The process of collecting the DNA fragments with the help of a glass rod is called as spooling. Now, the DNA is cut into fragments by the action of restriction enzymes. Now, gel electrophoresis is performed to check the progression of restriction enzyme digestion. We add the stain ethidium bromide and we visualize the gel under the UV light so that the fragments will be visible as bright orange colored fragments. Now, we will see few previously asked questions from the topic. So, the DNA fragments separated on agarose gel will be visualized after staining with. So, what is the stain that we use? We use ethidium bromide as the stain. The next question, DNA fragments are. We discussed since DNA has phosphate groups in it, DNA will be negatively charged. Now, what is the criteria for DNA fragment movement on agarose gel during gel electrophoresis? We discussed larger fragments will be closer to the wells, closer to cathode. Smaller fragments will be away from the wells and they will be closer towards the anode. So, negatively charged fragments do not move is the wrong option. Larger the fragment, the slower it will move. So, this is also wrong. The smaller the fragment size, the farther it moves. Yes, this option is correct. So, this is the third option. DNA precipitation out of a mixture of biomolecules can be added by treatment with. So, to precipitate the DNA, we add chilled ethanol. So, it's option 2. During the purification process for recombinant DNA technology, addition of chilled ethanol precipitates. So, in previous question we saw that chilled ethanol is used to precipitate out the DNA. So, it's option A. When DNA stands on gel stained with ethidium bromide, when viewed under UV radiation appear as. So, what is the color of the fragments that we see? We see them as bright orange colored bands. Next question, there are two statements which are given. Statement 1 states that the DNA fragments extracted from gel electrophoresis can be used in construction of recombinant DNA. The last step we discussed, after elution, the DNA will be used in cloning with the vector. So, statement A is true. In statement 2, smaller size DNA fragments are observed near the anode while larger fragments are found near the wells in the agarose. This is also true. So, both statement 1 and statement 2 are correct. So, it's option number 2. The next question, which of the following statements are correct with respect to DNA separation, isolation and visualization? The cutting of DNA is done by molecular scissors. Yes. The DNA fragments separate according to their size in the agarose gel upon electrophoresis. Yes. When electric field is applied, these fragments separate according to their size. The separated DNA fragments can be seen without staining, and when exposed to UV light, this is no. Even though we are observing it under UV light, we need to add the stain ethidium bromide. So, statement C is wrong. The separated DNA fragments can be seen without staining when exposed to UV light. No, we need to stain the DNA with ethidium bromide. The separated DNA fragments when stained with ethidium bromide can be seen in visible light. No, still you need to see it under the UV light. So, statement A and B are correct and statement C and D are false. They are asking for the correct statement. So, it's option number 2. Which of the following statement is not true regarding the gel electrophoresis technique? Bright orange colored bands of DNA can be observed in the gel when exposed to UV light is true. Second statement, the process of extraction of separated DNA strands from gel is called as elution. This is true. The separated DNA fragments are stained by using ethidium bromide. This statement is also true. The presence of chromogenic substrate gives blue colored DNA bands. It is false. It is orange colored DNA bands. So, it's option number 4. The same question is repeated again. If you see, DNA strands on gel stained with ethidium bromide when observed under UV radiation, they appear as which color? Bright orange color. So it's option 4. So the last question, a linear DNA molecule of 10 kb has 3 restriction sites. After digestion, we get 4 fragments. One is of 2 kb, 3 kb, 2 kb and 3 kb. So what is the first question that they are asking? How many fragments are produced? So in a linear DNA, there are 3 restriction sites. So the number of fragments which you get will be 4. First fragment, second fragment, third fragment and the fourth fragment. So how many fragments are we getting? 4 fragments. So if you see the second question, they are asking how many bands will appear on the agarose gel. Remember, bands and fragments are different. Fragments are the number of pieces which you get. So how many pieces of DNA we are getting? We are getting 4 pieces of DNA. But bands, if the fragments are of same length, they will migrate to equal distance. So they will be seen as a single band only. So here we have 2 fragments of 2 kb length and we have 2 fragments of 3 kb length. So both the 2 kb fragments will be seen as a single band and these 3 kb fragments will be seen as a single band. So the total number of bands which you get will be 2 bands. So fragments will be 4 fragments while the bands will be 2 bands. So these are the different steps of recombinant DNA technology. Thank you for watching. Please like, share and subscribe to Aakash Telugu YouTube channel.